everyone, and thank you for joining us. Let me first introduce RedTalks. RedTalks is a webinar series dedicated to open innovation in the field of lymphoid malignancies. These are short format episodes designed to bring together leading voices from academia and industry and explore most pressing scientific and clinical topics in our field. So for today's episode, which is the third out of fifth session, we are delighted to welcome Dr. Parikh, who is oncologist and director of the HACLab at Emory University in Atlanta. He will be discussing a particularly interesting topic, which is about generalizability of oncology trial results to real-world patients. I have to recall you first before letting him speak that you are able throughout the session to ask questions using the Q&A box at the bottom of your screen. And after the presentation, we will address them.
Okay, so I'm very pleased to welcome today's speaker. Dr. Parikh is an oncologist and the director of the Health, AI, and Cancer Lab at Emory University. And his research focusing on real-world evidence and use of machine learning methods to bring the gap between clinical trials and patient populations. And now, Dr. Parikh, I let you the floor.
Great. Well, thank you very much, Dr. Latouche and Dr. Bharat, for the introductions. And it's fantastic to be speaking with everybody. I'm going to try to go through in about 30 minutes some work that we've done around trying to facilitate generalizability of clinical trial assessments and talk through some of the methodology and motivation for doing so. And then hopefully we'll get to spend about at least 20 to 30 minutes discussing some questions and ways that we can either improve upon the work or extend the work into directions that are applicable for lymphoid malignancies. So here are my disclosures. So maybe just to start a little bit, I'm hoping to just give a bit about our lab, which is, as mentioned, called the Human Algorithm Collaboration Lab, or HACLab. And so we're broadly a lab that focuses on applications of big data and data science and oncology care. About half of our portfolio is geared towards trying to develop innovations applicable to cancer clinical trials. And so when I say that, I mean trying to develop AI or predictive solutions to try to, say, for example, predict patients who may be likely to be eligible for a cancer clinical trial, or try to extract certain pieces of trial-related information from unstructured clinical text, or trying to design so-called digital twins, sort of computational phenotypes of patients that could be used as part of prospective trials in the future as control arms or intervention arms as in clinical trials. But we do have a portfolio outside of this clinical trial infrastructure portfolio, and those include trying to develop methods to build trustworthy AI, namely to build AI tools that are less biased and more reliable when used in clinical care. We do have some prospective trials of AI-based decision support tools that we run. And then we have a portfolio relating to health policy and AI that I think is particularly interesting and only starting within the past few years, that largely focuses on how we can try to explore and redesign different types of policies, both in the US and more broadly, around use of AI in clinical trials and in clinical decision support. But I'm going to focus a little bit on a particular project that we have worked on that I think has applicability for the interpretation and future design of clinical trials. So that's this project, and I just want to acknowledge the first author of this paper, Xavier Orcutt, and then my collaborators, Ronak Mantani, and my co-PI, Chi Long. So I think it's before going into the details of this analysis, it's useful to talk about trial emulation, or the process of trying to emulate a clinical trial using real-world data. And so one question to ask just from a clinician standpoint, from my standpoint, is, why would we want to do this? And I can think of sort of a few different reasons. One is that we might want to assess the generalizability of a clinical trial. There's a clinical trial that was done applied to a real-world population. Another potential might be to compare two treatment arms that can't reasonably be compared in a clinical trial or are unlikely to be compared in some time in the future. And so say, for example, there are two standard of care regimens, and it would just take too long or be challenging to run a trial to compare those regimens. Then you can think about a real-world emulation of those two agents. And then the other might be, once an agent is approved, can we try to simulate alternative dosing schedules or follow-up frequencies as part of a given regimen using our real-world data in a way that might be difficult to assess prospectively. So when we think, hopefully that's convinced you a little bit about the motivation for trying to emulate a clinical trial. And there, as I mentioned, there's a lot of applications, both pre-trial and after a trial is done, that we might want to do this kind of thing. But so when we actually talk about emulating a clinical trial, what do we actually do? So the first, I would sort of think of it akin to how we design a real, an actual clinical trial. And I think the highest quality trial emulations try to mimic as much as possible from a real-world clinical trial in terms of eligibility criteria and protocol design and et cetera. And so if you think about a comparative effectiveness question that you might have, comparing one regimen versus another regimen, for example, if you were going to design a clinical trial protocol, you would write down all sorts of elements relating to eligibility criteria, treatment strategies, how you're going to assign procedures to somebody, what the randomization schema is, for example, your follow-up period, your outcomes, and then your analysis plan. And I think the highest quality emulations try to specify exactly that. They try to, basically come up with a lengthy protocol that specifies all of these elements and just have that applied to a real-world data source. And I see this as a little bit different than our typical comparative effectiveness study, where we might have a causal question in mind, but we might not go through the steps of actually stipulating a protocol and doing that before we run the study. And so I think that's what sort of differentiates, or is a major differentiator of a true trial emulation, is actually pre-specifying what you want to do. And therefore, we can interpret the trial emulation with the same type of fidelity as we might a clinical trial. So now that we've talked a little bit about the motivation behind trial emulation and how we think about doing it in a high quality way. Now let's talk a little bit about generalizability of clinical trials. So say that there is a seminal publication that comes out in the Lancet or the New England Journal of Medicine or BMJ or whatever it might be. And I'm very excited with this publication, but now I have a patient sitting in front of me and I have to decide, do I change my practice based on this clinical trial? Well, when I look at the clinical trial, normally what I'm faced with are things like a survival curve, like overall survival of one agent versus another. And then I look at a forest plot or some sort of heterogeneity of treatment effect assessment. And those sort of tell me what I might expect. Now, one of the challenges is that cancer clinical trials oftentimes have somewhat restrictive eligibility criteria. And even if their eligibility criteria are not restrictive, there might be certain reasons why someone gets enrolled in a trial compared to a general real-world population. And so, typically our heterogeneity of treatment effect estimations, these forest plots, they're not too revealing for how I ought to be deviating from this average treatment effect that's published in a trial, because these forest plots, these heterogeneity assessments, are only usually based on one variable versus another. So you can see for age, it's less than 65 or greater than 65. And then you try to repeat your point estimate and confidence interval. And so this is challenging, I think, because I don't think about my patients as less than 65 or greater than 65. I don't think about a patient sitting in front of me as ECOG 0 or ECOG 1. I think about them as an amalgamation of different factors, some of which might have better predictive effect and some of which might have less of a predictive effect. And so it becomes really challenging for me to extrapolate from these forest plots to making a decision about whether I should treat my patient in front of me different than what has been stipulated in the clinical trial. And I think that is what sort of motivates some of this generalized, more advanced methods to try to develop better estimates of generalizability. Now, why is trial generalizability even a problem? Well, as you can see from the graph on the left, which comes from a JCOP paper published in 2021, patients who are well represented in clinical trials, indicated in blue, tend to have much greater survival than patients who are unrepresented or underrepresented in clinical trials, indicated in the red and the green lines. And so some of that, again, has to do with restrictive eligibility criteria, but a lot of it also has to do with, there might be certain factors that, for example, transportation access or, all sorts of other factors that result in a patient being offered a trial versus not offered a trial. And so that poses a real gap when it comes to trying to generalize results of these highly selected clinical trials to a broad real-world population. Now, there have been efforts to try to do this. So this is a paper on the right here that was published by James Zhao's group out of Stanford in 2021 that showed when in the real world, if we were to relax certain eligibility criteria in the using real-world data, then oftentimes we're able to still preserve a hazard ratio or a treatment effect that's seen in a trial. And that this paper, among others, has been used as a lot of justification for trying to relax some of the restrictive eligibility criteria that we have in trials to make some of our trial results be more generalizable to a real-world population. Now, when you look at the sort of prior efforts at assessing generalizability of clinical trials, a lot of them have just focused on whether, in the real world, you can sort of recapitulate a clinical trial effect. So this comes from a study that was published out of Sebastian Schneeweiss's group in JAMA that showed that, in general, when you apply good confounding control methods to real-world data, you're generally able to preserve somewhat of a treatment effect that's seen in the real world in a clinical trial. But that, I would argue, that's not necessarily what I'm most interested in. I'm not most interested in seeing whether you're using a fancy set of statistics and curated populations if you're able to preserve a trial effect. I'm interested in learning whether a trial is generalizable for even my sickest of patients or not. And if not, what are the characteristics of patients whereby I can safely generalize a clinical trial versus where I can't and where I might have to make a decision that deviates from a typical clinical trial. And so that's largely the effort that we tried to undertake in this paper, which is, I think, the subject of this talk. And I think about it, the title of the paper is published in Nature Medicine earlier this year. And the title is Evaluating Generalizability of Oncology Trial Results to Real-World Patients Using Machine Learning-Based Trial Emulations. But I kind of think about the goal of this paper as being trying to estimate, trying to develop computational representations of the average patient that walks into my clinic, and then trying to explore for what types of patients cancer clinical trials generally, approximate pretty well versus which ones they don't. And so we used to investigate this question, we took a cohort of patients with several types of advanced solid malignancies, so no lymphoid malignancies in our particular data set, although I think the platform is easily generalizable to that. But we had a data set that consisted of about 2 million patients from over 280 clinics in the US that were primarily community oncology based. And those data from those patients were linked, they consisted of electronic health record data and curated registry grade cancer-specific information on things like stage and performance status and treatment receipt. And then we linked that data set with data from commercially available next-generation sequencing panels from a company called Foundation Medicine, which is one of the foremost next-generation sequencing providers in the United States. And so, we had this very rich, granular set of clinical genomic data to be exploring these type of trial emulation questions, as well as very high-quality, curated progression data and mortality data. So here was our approach. In the first stage of this pipeline, you can see from the top here, we first took data sets within each of the 4 advanced solid malignancies that we were working with. In this case, we chose to work with data sets that had relatively large sample size, so breast, colorectal, non-small cell lung, and advanced prostate cancer. And so within each of those disease-specific data sets, we first split the data set into training data sets and testing data sets. And from those training data sets, we first extracted a whole host, almost 500 variables that was based on longitudinal EHR or genomic sequencing data. And then we used some standard techniques to impute missing data, after which we fit a series of machine learning-based models you can see indicated in the green and purple starburst icons here that serve to predict the likelihood of mortality at the start of first-line therapy for advanced disease. So that was what we used to develop an algorithm. From that algorithm, we further phenotyped. Then we moved on to the trial emulation component of our work, where we actually tried to emulate eligibility criteria for those trials, sometimes in a more relaxed manner, only focusing on the specific type of cancer and treatment, and then correct biomarkers. And then in a sensitivity analysis, we actually more strictly emulated those studies using more granular biomarker-based variables and other more granular clinical variables. But once we came up with a cohort that sort of met eligibility criteria for a given trial, then we applied our best performing machine learning model, indicated in the graph on the top, to try to phenotype patients within those cohorts into high, intermediate, and low risk prognosis groups. And then once we did that, we applied some relatively standard propensity score methods to try to estimate the time from start of first-line therapy to death using Kaplan-Meier plots within each of these disease-specific phenotypes. So I'll go into some of the specifics behind the methodology for all of this, but that's essentially, this gives a pretty high-level overview of what we did as part of this machine learning-based platform. So as I mentioned, the first phase was phenotyping, and I think I went through the steps that were involved here. I think one important point to note is that in real-world data, there tends to be a relatively high amount of missingness, either because certain electronic health records aren't capturing all the data that's available as part of a broad data set, but also because some patients might receive a certain lab and some might not, or some people might receive a certain type of visit and some might not. And so oftentimes we have to impute missing variables to allow these models to function. So we used relatively standard imputation models to try to ensure a lack of missingness in our overall data sets here. From then we went on to fit some of our models, and you can see an example of one of the machine learning models that we fit here, whereby on the graph in the green and the graphs in the green, we saw that our gradient boosting models, which is a type of supervised machine learning model, generally outperformed other machine learning and non-machine learning-based models at predicting risk of overall survival. We furthermore looked at which variables were most important in that prediction, and many of these variables were akin to what would be normally thought about as prognostic in a real-world setting. So we did this across all 4 of our cancers, and you can see that even though some of the models, there were different variables that ended up being more versus less important between different cancers, overall, our machine learning models, particularly our gradient boosting models, tended to be the best performing models for predicting someone's prognosis. So that gives some insight into phase 1 of our process, which is machine learning informed. In phase 2 of the process, we actually tried to emulate these seminal clinical trials that we were looking to emulate within each of these 4 diseases. So we matched a set of real-world data based on data, in restricted patients who had an eligible type of cancer, who received a guideline-based therapy, and then who had the right biomarkers that were studied as eligibility criteria in this study. And so once we did that, we used our machine learning model that I talked about before to try to risk stratify patients into high, intermediate, and low risk of experiencing a progression or death. And then once we fit those models, we then ran them and used them to risk stratify the population into those 3 groups, after which we used the propensity score methods to actually run a retrospective clinical trial. Now, someone might ask, well, how did you arrive at which trials and which cancers to focus on? So as I mentioned, we focused on the 4 highest represented cancers in our data set, lung, prostate, colorectal, and breast, mainly because for these models to get reliable, they generally need a lot of data. And so we wanted to make sure that we had adequately represented cancer sources. But there's no reason why a well-represented lymphoma or myeloma data set would not also be applicable for this platform. And then, so once we identified the relevant trials, we restricted them to phase 3 clinical trials, which generally involve some sort of comparative assessment. And then we looked at, of those trials, which ones had results that were available that we could compare to. Once we had that, then we looked at the available study protocol to be able to see whether we could emulate the eligibility criteria. And then we furthermore ensured that there was adequate minimal sample size within our data sets, which is how we arrived at 11 trials across all 4 of these malignancies that we chose to emulate. Some people might ask, well, it's really hard to balance observable covariates with propensity score-based methods applied to real-world data. And as you can see on this graph here, while there were some baseline differences between our treatment groups, as you would expect in the real world, like certain groups might be healthier or have lost less weight, whatever it might be. In general, when we applied our propensity score methods, most of our variables were quite balanced and not necessarily too discrepant. You can see that by the fact that the blue adjusted dots generally tend to fall within a 0.1 standardized mean difference, which is a common threshold that's used to assess whether your trial is well balanced on certain observable characteristics. And then we ran our trial emulation. And what that largely showed was that you can see that the top line sort of finding on the bottom here is that clinical trial absolute and relative survival estimates were maintained for some of our highest prognosis or low risk individuals. But for lower prognosis, high risk individuals, generally both the absolute and the relative value of an experimental combination or agent in clinical trials generally wasn't found. And so I'll dig into a little bit about what that means. So here's a representative example from a seminal lung cancer trial, Keynote 189, where we ran our trial emulations. And so the way to interpret this graph, so shaded in the red and the pink are the point estimates and 95% confidence intervals from the seminal clinical trial that was done, this Keynote 189 trial. This is what was published in the New England Journal of Medicine. In our emulations, you can see those represented by the black and white dots. So the black dots represent the intervention arm, which in this case was pembrolizumab with chemotherapy, versus the control arm, which was generally chemotherapy alone in this trial. And so these point estimates, these dots here, are the results of our trial emulation, where they represent the average overall survival in our low, medium, and high risk groups. And so I think the way to interpret this is that the dots tend to be farther apart, meaning that there's an effect of the intervention arm in both the low and medium risk prognosis groups. So those are groups where generally the finding from the clinical trial is preserved. Now, the magnitude might be smaller, the magnitude of survival difference, but the general finding is still preserved. But for our highest risk groups, these are the poor prognosis individuals, generally we found very little absolute or relative survival benefit from, in this case, pembrolizumab compared to the standard of care. And so this high-risk population, which we found across different cancers, generally represents a group for whom clinical trials generalize somewhat poorly. And I think the benefit that we're offering from this platform is being able to predict which patients might be at highest risk of not only passing, but also not resulting to a clinical trial the way that we think it should. We did do some robustness checks to assess the quality of our emulation. So I won't go through these factors, but basically we ran a bunch of checks on all of our studies to see whether they met some standardized sort of quality control criteria for trial emulation studies. And lo and behold, about half, maybe a little bit more, did meet those criteria. But I think it just serves to show that there's probably a lot more to go in trying to ensure high quality, high fidelity of the actual emulation that we're trying to do here. This is sort of a summary level finding where we aggregated data from across all of the trials that we studied, the 11 emulated trials. And we just plotted how different was the estimated survival in these prognostic groups compared to the clinical trial. So what you see on the y-axis is the estimated difference in the survival difference or survival benefit from the real world from the clinical trial to the real world data. And so the way to interpret this is if the survival benefit, meaning the difference in survival between the intervention and control arm, if that was the same as in the clinical trial, then the difference between the two would be 0. But what we generally saw was that real world survival benefits were generally a little bit lower in the real world population compared to the clinical trial, indicated by mostly negative findings on this box plot. And that the magnitude of how far off we are, how far different our emulated trial result was from the clinical trial, that magnitude went much higher as you went on in increasing levels of risk. And so for the high risk group, there was about a 7-month survival difference or survival benefit difference in the high risk group compared to the overall clinical trial. And what that tells me is that this is generally a population in whom trials don't generalize all too well. We also did some robustness checks, additional robustness checks. So instead of just matching on age and stage and organ function, we actually also tried to match on more granular eligibility criteria for trials, namely biomarker-driven eligibility criteria. And what we generally found here was that most of our relative and absolute effects were still preserved when we did a more strict emulation or strict eligibility mapping of our of the patients in the real-world data set. There was also a critique that we got that maybe one of the reasons that your results are different is because in clinical trials, they generally have standardized dosing paradigms, but the way that you dose in the real world might be very heterogeneous. Like, sometimes I'll start people pretty early on a reduced dose of a standard of care, chemotherapy or oral therapy. And so we accounted for this because even when we only analyzed patients who had not de-escalated therapy, who had consistent either oral or IV therapy throughout their treatment journey, even when we restricted to that cohort, we still generally maintained the real world results that we found. The estimates were not all that different. I won't go into this, but we did several other statistical robustness checks to try to sort of just maximally interrogate all of these findings for these high-risk individuals. And we generally found that even when we use some of these statistical differences in our paper, we still generally found the same effect. So one question that at least I answered, and I would hope that you're asking right now, is how does this eventually, could it be used in clinic? Like, what does it matter if we identify patients who might not be generalizable from a clinical trial result. And so we've actually formulated our algorithm into a web-based calculator whereby you could plug in variables for a patient and get not only what risk categorization they're on, like high, intermediate, low risk, but we can also get to a personalized emulation of a trial in someone like you. And so on the next slide, you can see how that looks if people plug in the requisite information. And they generally get a notification that they're either high, intermediate, or low risk. And then they can see what is the sort of real world survival patterns for patients like yourself who are getting the one of the two drugs that are in question. I think this is not, research. This is research grade. This is not sort of validated by the, some sort of regulatory body, but we meant it more so to sort of highlight that we're interested in translation of these findings. And, this offers an opportunity, this sort of calculator offers an opportunity, if it's scaled, for people to be using this to get individualized risk assessments for their patients. So maybe those were the ultimate results of the paper that was published in Nature Medicine. I wanted to maybe take a step back and just talk about some of the implications of these findings and what we're really excited about now. So, I would sort of characterize our work as being sort of emblematic of a digital twin. We've created these sort of algorithms and computational representations of patients. And so, one of the use cases of digital twins that's been put out there is to do exactly what we did, translate clinical trial evidence for a real world population. But there are other ways that we could use a model like this. Like, for example, we could use it for more direct clinical decision support. So, for example, could you use this in real practice to sort of identify which patients are more or less likely to look like the patients that were in the clinical trial? We can also use these as these computational representations as control arms in clinical trials, as you can see in the top left. And then there's a variety of other AI-specific strategies that I won't go over, but just say that, like, the advent of these technologies has made the prospect of using these so-called digital twins that we've developed, these computational algorithms that represent real-world patients. They've really opened up the door for these to be used in a whole host of other potential use cases. So, how can we move from generalizability assessment to actual better trial design? So, one of the things that has been brought up in other published literature is the fact that if you have an algorithm for a patient that can be applied to some sort of real world data, then technically you could have an external control arm for a phase 1 or phase 2 study that could give you a general degree of confidence in your survival estimate. And so this has been traditionally absent from phase 3 trials in the US because most of them don't do randomization. So, we've been really excited about the idea of trying to use some of the models we've built to help inform external control arms for these sort of in silico trials, but that's a bit early. So the main takeaway is that machine learning-based trial emulation can reveal real-world heterogeneity in clinical trial design and clinical trial generalizability assessment, and could be used to help inform future clinical trials if, say, for example, we identify that a particular treatment doesn't work for a certain subgroup. Could that subgroup be subject to a different type of clinical trial? So I'll just close in saying that, we are interested in extending this work. So we have, largely informed by some of our experiences with this past paper, we've been trying to undertake an effort to map eligibility criterion onto standardized eligibility queries so that hospitals and health systems can more reliably estimate how many patients and what sort of proportion of patients are truly eligible for a trial, and how can we sort of increase that. We've also taken an interest towards trying to emulate negative clinical trials that didn't show a benefit of intervention versus control, to help say, well, maybe we're able to identify certain phenotypes of patients, just like we did in the study above, that may respond better to the drug, even when the average treatment effect is not too significant. So those are just two general ideas out of a few more of sort of active projects that we're thinking of working on or have worked on. A lot of our work has been informed by the collection of high quality annotated trial data. And so we've tried to do this, and this is all multi-cancer data. Heme malignancies are pretty well represented in all of these data sets. And so part of our goal has been to aggregate these data together to help serve as some sort of data lake for future types of trial emulation type experiments or other types of experiments regarding to predictive model development. So with that, I'll just close and thank you all for the time. And I think I'd really love to engage on whether this work is sort of relevant to your related research and how we might think about extending some of the work to lymphoid malignancies. So with that, I'll stop talking and thank you all for the opportunity.
Okay, thank you. Thank you very much, Dr. Parikh.
Thank you very much.
Now it's time for your questions. So I have to recall you that you can ask them and submit them via the functionality Q&A. And we already have 2 questions. Congratulations from Clémentine Sarkozy. And then another question about performance status.
Yes. So this is a great question. Can I take it?
Yes.
Okay, sure. So the question is, certain factors like ECOG performance status is heavily missing in both the database we looked at as well as all real-world databases. And given that the missingness may not be random, since sicker patients are often less likely to have ECOG, how did you assess and mitigate the risk of bias that this could introduce? So this is an excellent point. There have been, a lot of quality improvement efforts, including at institutions of mine, that have been trying to just incentivize doctors to better code things like stage and performance status. Now, this poses a real risk for studies like ours, because if data is missing, we don't know whether standard imputation-based methods, which normally assume a missingness at random, whether those are actually going to apply or not. The advantage that we had with our data set, and I think is true across others, is that we actually didn't have a lot of missingness among patients who met standard trial eligibility criteria and who received the treatment. We didn't have a lot of missingness in performance status. Those missingness rates, which are generally about generally about 40, 50% in a general population. Once you curate them down into patients who have sort of minimal data standards and who have received treatment, usually the performance status missingness rate is somewhere on the 15 to 20% range. And from there, it's generally accepted that imputation at that low level of missingness is a reasonable strategy to think about, but it does presume that this missingness is going to be at random of some sort, and it might be that patients who didn't have their performance status collected may have come from certain localities or other domains. So we in some sensitivity analyses, we also used in addition to different imputation strategies, we also used a missing indicator strategy just to indicate, just to try to flag whether there may be certain factors that might result in missingness or not, and trying to sort of account for that using a missing indicator strategy. And in general, we still found similar results. I don't know whether those results are going to be the same if we went to a completely different data set that didn't have as good curation of performance status or stage or things like that, because then there becomes a real risk that you're trying to impute something without accounting for this sort of missingness, not at random kind of phenomenon. So hopefully that answers the question, but we did think very hard about that.
Okay, thank you. And we have another question about the risk group. I think it refers to the machine learning prognosis score. And does it take into account the treatment effectively received by the patient? And in this case, could the risk category that is assigned to the patient depend to the treatment received and thus interfere with the emulated trial step that comes after?
Yeah, so this is a great question. So just to answer the first part, we did not account for treatment received in our predictive models here, because that would have involved data leakage, when you use future data in your predictive models. So we did not account for that. Now, there's still a potential bias in these real world models, because even if you don't allow data to leak, your outcome of interest might be influenced by the type of treatment that you received. And so to account for that, we tried to use inverse probability of treatment weighting to account for the propensity to receive a given treatment in some of our predictive models. And so in general, as you could see, we were able to balance on observable characteristics. I'll show the slide again that demonstrates that. But we were generally able to balance on observable characteristics when we did that inverse probability of treatment weighting. And so that led us to be a little more reassured that we were accounting for this, this heterogeneous treatment assignment. Yeah, I think the learning lessons here are probably one, that we have to be very careful of not allowing data leakage, because we were very strict in only allowing data from at or prior to the start of first-line therapy to be included in our models. But so we have to be very careful in some of these other analyses that data leakage, either intentionally or unintentionally, isn't happening. Otherwise, it becomes very difficult for this to inform prospective treatment design. But we think that this actually could be used to inform prospective treatment design, given that we tried to account for that. So, imagine if, say, for example, we treated this, our platform here as some sort of like correlative biomarker, where you were running a clinical trial and then could observe treatment effects within these sort of buckets of high, intermediate, low prognostic risk. So that would be one envisioning of how this could be incorporated in future trial design.
Thank you. And then you mentioned the possibility of your tool to drive and help designing future clinical trial and randomized trial for novel agents. And is there not a possibility of data leakage between your initial estimations and prognosis score that is developed in your paper and the possibility to use this data to design other clinical trials?
Yeah, I think that's such an excellent question. So here's a couple of ways we've thought about it, but I'd love to sort of have it back and forth with others about whether there's other ways. So one, maybe just taking a big step back, like we're doing this all with retrospective trial data. We're not doing this with prospective trials. We're not doing this with agents that haven't yet come out on the market. But say, for example, you ran our study and you can see that there's a certain subpopulation of individuals who don't really benefit from some sort of novel treatment regimen. Maybe there's a certain subset of individuals who really don't benefit from the addition of pembrolizumab. Well, one way that could be used to inform future treatment design is to focus on those subgroup of individuals that don't respond, like typical clinical trial populations do, and to focus future trial designs on that population. And maybe the type of trial that we design in that standpoint is more so about, trying to use reduced dose or different intensity of certain therapies and try to test a trial in that way. Or maybe we try to test a de-intensification strategy where we take some sort of new standard of care and try to de-intensify it among this poor prognosis group to see whether that results in better outcomes. So those are some ideas about how it could be used to inform just standard trial design. And then this idea of trying to design, evolution-based trials where you might use this type of phenotyping to refine the type of therapy that someone gets down the line. So maybe there's some sort of interim response assessment, and then you use that response assessment to sort of intensify or de-intensify or assign someone to a different treatment regimen. That is a big possible use case of platforms like these, or other just AI-based platforms more generally. And I think that is sort of where a lot of this is heading, because I think sometimes we're just a little too fixed with our original trial design parameters.
Okay, thank you. And also you mentioned retrospective data only. Is there a possibility of your strategy to be evolving according to time to incorporate more and more data from real life and evolve at times goodbye to incorporate temporal trends?
Yeah, absolutely. So, for example, I think one really so these models are all built on real world data. And so it's relatively easy to retrain our models using updated data sources and try to use that to re-phenotype patients. So that becomes not a difficult thing to do, given that we've already built out the code for extracting data from real world data sources, and all of these real world data sources are continually ingesting data every quarter or every 6 months or whatever it might be. The other possibility, though, and this I think is particularly relevant in the lymphoid malignancy space, is given that some of our therapies for later line progressive disease have become really good with the advent of CAR-T and BiTEs and other types of therapies, you might even be able to use strategies like this to assess different types of sequencing of certain therapies. We encounter this a lot in my field, prostate cancer, where we don't know how to sequence chemo versus the androgen receptor signaling inhibitors versus other types of therapies, and all of them are approved in the first line setting. So we can use these type of trial emulations to identify phenotypes of people who might benefit from one sequencing regimen versus another, and then have that be incorporated into some practice guidelines. So that is absolute. I'm not sure if that fully answers the question, but I think that the ability to ingest new data is one of the real advantages of using real world data and these type of adaptive trial emulation type frameworks.
Okay, I will take now the lead for the next questions. Thank you. So a very naive question: can such technology be thought as a way to introduce parameter during trials and a way to optimize individual treatments?
Yeah, I think so. So, when you think about, just think about what we normally use as sort of biomarkers or correlatives in clinical trials or ways that we might fine-tune treatments. Maybe it's based on some sort of isolated biomarker, like tissue-based biomarker, like, PDL1 or EBV, CMV status, or whatever it might be. Or maybe it's based on some sort of crude risk score that's based on clinical factors. I think the advantage of these machine learning-based parameters is that they can incorporate both clinical factors, biomarker-based factors, and also just interactive relationships between all of those factors. And they can actually come up with holistic risk scores that I think are just much better risk stratifiers than our typical like univariate or unimodal risk factor assessments. And so if you could envision using this as, in a trial to either, identify prognostic phenotypes to assign people to differential treatment strategies, I think that would be really important and a really, cool use case for prospective trial design. I think the key is, though, that you have to move from just this prognostic phenotyping, which is where most machine learning-based papers stop. They say, okay, are you high risk for certain diseases or low risk for certain diseases? And then they say, okay, we've built this prognostic biomarker or this correlative. And you might say, oh, you have to go from that and actually try to investigate whether differential management in the real world, using these real world emulations, does differential management strategies actually, are they beneficial? So is it beneficial or non-inferior to use dose-reduced chemotherapy among poor prognosis individuals or not? And so if you can test that in your retrospective data and show that your AI biomarker is actually predictive of benefit of some sort of alternative treatment strategy, then I think you can take that into a clinical trial and actually use that for some sort of treatment assignment type of thing. I think it becomes a little risky to just presume that, okay, just because someone is poor prognosis, they're not going to benefit from high intensity chemotherapy. You actually have to assess that in your real world data. And that's kind of what we tried to do in this study.
Okay, thank you. Yeah, to go ahead with these questions, I have one question about you use the tertiles of the risk distribution of the risk.
Yes.
Do you have the risk I did not find the risk shape distribution?
Yeah.
How is it looks like?
Good question. So this is a really important point, especially for certain cancers that have like compressed risk distributions, like early stage malignancies, for example, because some sort of crude binning, you're sort of treating someone who has, 49th percentile as different than someone who has 51st percentile, when in actuality, they have like basically the same risk. When you look at the risk distribution for this metastatic population, in general, about 50% of patients die at 1 year. And we used 1 year as our training point for these models. Our 1 year mortality is our training point. So we had relatively bell curve shaped risk distributions. And this is living somewhere in either a supplemental figure or somewhere that we didn't include in the supplement, but we have data around. So we felt comfortable using a tercile-based strategy, but it is sort of an arbitrary binning strategy. And even though we use terciles, there might not be a lot of difference between someone who's at the 32nd versus 34th percentile. And so I think we do need to use these type of strategies and apply some more clinically informed thresholding, because that becomes a real risk when you treat someone high versus low different. I'll just say though that I encountered the same problem when I encounter if I treat lung cancer, we use PD-L1 50% as sort of a threshold. And I have no idea as to whether PD-L1 49 versus 51 is any different. But I have an FDA-approved indication to use something greater than 50, and I don't for using something less than 50. So I think these threshold-based things we have to figure out across all of oncology, and not just for this machine learning stuff.
Okay, thank you for the answer. Now I will move to the question about the divergence in emulated overall survival across risk group relative to clinical trials. Raise the question of whether the GBM model might be over-smoothing, potentially masking important signal variants, especially in high risk or less represented subpopulation. Could you comment on this possibility?
Great point. So I think this question sort of is alluding to the possibility that certain models, particularly the GBM, are overfitting on certain subpopulations. And I think it is a really important point that I don't have a great answer to, that we did not do, while we used the standard train test split for building our models, so we had a holdout set within our overall population. We don't know whether our models are overfit to the Flatiron population in general, and whether they're going to generalize to some other sort of real-world data set. I think we have general sense that the Flatiron data are representative of a real-world population, and that the representation of certain populations are akin to what we would see in a typical SEER Medicare population. We cited some previous data to that effect. But as to whether the data is going to be overfit for less well-represented subpopulations, that is a very open question that I don't have a good answer to. I think it requires, though, a little bit of thinking through. Just because some population is underrepresented in one of our data sets, does that mean, do we have some sort of biologic plausibility as to why that population would respond differently than the majority population, or differently than our prognostic phenotypes would? And I don't know if I have a great answer to that, because sometimes I see these critiques as to, your representation of rural patients is different than urban patients, or your representation of certain minorities is different than other populations. And my question back to them is like, is there some sort of biologic or socioeconomic reason as to why you would expect this patient to differ in terms of treatment response compared to a majority population? And if you don't, then I don't think we could go ahead and assess subgroup performance for everybody. I think we have to have some sort of hypothesis-driven rationale for thinking that these models might be less accurate for certain subpopulations. That's just my opinion on it.
Okay, thank you. Yeah, to go, along with this question, I have a small question. You consider the best model among a collection of models, and I was really expecting when I I was asked to read your paper, give the opportunity to find some ensemble methods such as superlearner to have an optimal combination. I also realized that it would add a top layer on top of something which is already complex. But do you think it would be doable in the midterms to have some doubly robust approach?
Absolutely, and I think so ensemble-based models that kind of integrate some of these different some of the different models, I think, is a great direction to take this. We also did an experiment with like deep learning-based, more black box-based methods that equally might be interesting here. I sort of think about this paper less of a machine learning paper and more about like a like a trial emulation paper. And so I think we're layering onto that trial emulation some sort of data-driven risk stratification that assesses heterogeneity. So I'm not quite sure we would get because all of the top identified features were all relatively clinically expected features of poor prognosis. I don't necessarily think that the top level, like certain patients might be categorized into different groups if we used a different algorithm-based stratification. And maybe we would have seen more of a widening between the terciles of our treatment effects. But I don't think the top level findings that there's just a lot of heterogeneity in real world treatment effects would have been all that different, because we're generally seeing these with relatively nowadays kind of stupid AI type models that we used in our paper. And so I think my expectation is that if we used a superlearner or deep learning based method, we would largely see the same flavor of findings with some refinement as to which patients fall into which risk buckets.
Okay, thank you. Maybe the last question. I don't know if we have more time, but I think this one is interesting as well. Given that your models were developed using US-based data, have you explored or planned any external validation using real-world data from non-US population?
Yeah, so we actually just had I just gave a talk and have just been exploring a collaboration that at a very base level would involve some sort of validation of this tool, but at a high level would hopefully involve something bigger. With a European-based data set through a group called Digicore, which many of you may have heard of. And I think more broadly, we are very interested in validating the findings, but two, deriving similar models in other types of cancers, and maybe we just have higher quality data in other cancers from other types of European-based data sets. So I would say it is a huge interest of ours, and we haven't done it yet, but with Digicore and others, hopefully we'll be able to do so, and would love to collaborate on that.
Okay, thank you. Do we have more questions?
Yes, maybe I can ask one. In your work, you showed that results from clinical trial cannot be replicated, or at least fully replicated, in observational data. And we are more used to result from the opposite. I mean, result from observational data not being replicated in clinical trials. So I'm thinking of the reason that may explain this phenomenon, and I'm particularly thinking on pragmatic trials. Pragmatic trials are usually usually designed for trials that leave more room for investigators for treatment, and also that have broader eligibility criteria. Are there any pragmatic trials in the trial set that you used? And do you think that, if not, do you think that your result may be altered if we are dealing with pragmatic trial, in which we expect maybe a result to be more reliable and more generalizable to a real world data?
Yeah, I think it's a great question. I think the whole motivation for doing this study is because the preponderance of trials that change our practice come from standard non-pragmatic trials that have relatively restrictive trial eligibility criteria. And so if we could get to a place where we did more pragmatic trials with looser eligibility criteria, frankly, I might not see a huge role for a paper like this, although even in a pragmatic trial with relatively looser eligibility criteria, you might still envision a scenario whereby there are certain phenotypes of patients that are underrepresented. I mean, when I think about who I choose to enroll in a clinical trial, there's all sort of non-eligibility related factors like, do they live close to my facility where they're going to have these standardized treatment assessments? Do they have distrust over clinical trials? All this other type of factors. And so I still think that there's a need to assess generalizability of clinical trials to real-world populations, whether it's pragmatic or not. But I think that pragmatic trials sort of throw out the eligibility criteria related reasons as to why a trial might not be generalizable.